Good morning, my little microbe lovers, microbiology students. Okay, we're going to talk about how to grow microbes. Yay! Microbial growth will increase the population of your microbes. So that's what you want. You want to put them in your incubator and you want their growth to increase. This is due to the reproduction of individual microbes, so you want them to be happy and reproduce and grow. Results of microbial growth should, if you're really good at it, result in a discrete colony, a little dot where you know that is the aggregation of cells arising from a single parent cell. So this is what we're going for. Eventually, we're going to streak some plates and we're going to hope to get a single colony, not just a wad of different bacteria, but a single colony. Also, you will get biofilm. We're going to talk a lot about biofilm in these chapters, in this section. Biofilm is a collection of microbes living on a surface in a complex community. So biofilm is like plaque on your teeth. Yay, you're going to want to brush your teeth after these lectures, just telling you. Organisms use a variety of nutrients. Different organisms use different nutrients. Now, I'm going to say this several times because I really want you to hear me. There is a difference between the nutrients required for a bacteria, which make it a selective media. So it doesn't grow on a certain media. So if it doesn't grow there, you know it's not that bacteria. That's a selective media. We use it for identification. There are other things that we know about nutrition that just make the bacteria grow better. So if you are the lab manager and you're trying to get the bacteria to grow, these are the things that it wants to make it extra happy. Those are two different things. Don't confuse this bacteria likes a thing. This bacteria likes nitrogen with it's a selective bacteria. It just likes nitrogen more. If you're trying to rear it in the lab, you're going to want to use nitrogen. But that doesn't mean it's a selective media in which it's going to tell you which bacteria it is. So don't confuse those two things. I will keep saying that over and over. Most common nutrients that are necessary for most bacteria: carbon, oxygen, nitrogen, hydrogen, and microbes out in the wild. Microbes in the wild obtain these nutrients from a variety of sources. Microbes in our lab maintain these from the growth plates or the broth that we use to rear them. There are two kinds. This should back it up to basic biology, right? Autotrophs, heterotrophs are the two groups of organisms based on the source of carbon. Then there's two groups based on the source of energy. Do you get it from chemicals or light? Chemiotrophs and phototrophs. I think I have a chart in here. Yes, look at that. So you have the energy source, in this case, plants, uses light, and they're autotrophs, right? So this is plants, green bacteria, things like that. Autotrophs, but they use chemicals. This is some archaea, some bacteria. Photoheterotrophs. These are green non-sulfur bacteria, and chemoheterotrophs. These are aerobic respiration, most animals, fungi, protozoa, bacteria, aerobic respiration, things like that. Chemoheterotrophs. Okay, you also have based on sources of electrons. So this is organotrophs and lithotrophs. Okay, now this one's going to be really important. So pay attention if you weren't paying attention before. Obligate aerobes and obligate anaerobes. So let's break that down for a minute. Obligate means you have to have it. So obligate aerobes mean you have to have air. You, as a human, are obligated to have air. Obligate anaerobes are you can only live when you are not in the presence of aerobic air. Oxygen. Okay, so oxygen is essential for obligate. You're obligated to be an aero. You're obligated to oxygen. Obligate anaerobes, you're obligated. You cannot grow in the presence of oxygen. Toxic forms of oxygens are highly reactive and excellent oxidizing agents. So this results in oxidation causing irreparable damage to cells that cannot grow in the presence of oxygen. So it is possible for things to be obligate anaerobes. Bacteria are sneaky little things. They have developed ways to survive. So Oxygen requirements. 4 toxic forms of oxygen: singlet oxygen, superradical oxygen, peroxide anions, and hydroxyl radicals. Many organisms can live in various oxygen concentrations. Aerobes uses oxygen as a final electron acceptor. Do we know this? Do we have to go back to cellular respiration? Right? You have all of the things that NADPH and glycolysis and all of that is making electrons, yes, that move the hydrogen ion concentration so that we can make the propeller that makes ATP. The key to that is your oxygens. And I mean, your electrons, and at the very end, you need oxygen to accept that hydrogen ion. It's the final electron acceptor. The final electron is accepted by oxygen. If you don't have it, it backs the whole thing up and there's no ATP production. So aerobes need that oxygen to be the final electron acceptor. Anaerobes cannot tolerate oxygen at all. No oxygen at all. They can't have it. Okay, now we're going to talk about facultative. Facultative is like with or without. I like it. I can do both. So they can live in various, so you can, some of the bacteria are like, I really would prefer to live in an oxygen environment, but I can live without it, or I can live with small amounts. That's facultative anaerobes. Aerotolerant anaerobes can tolerate only low levels of oxygen, and microaerophils require just a very low level. So these are just like varieties of different kind of anaerobes. Okay, facultative versus obligate anaerobes and aerobes. So if you were in class, I would make you like turn to your neighbor and tell me the difference between facultative and obligate, and then anaerobes and aerobes. Okay, so you can pause it and see if you remember these things. So an obligate anaerobe must not have oxygen. It's obligated to not have oxygen. An obligate aerobe must have oxygen. Facultative anaerobes can live with or without oxygen. Aerotolerant aerobes prefer anaerobic conditions, but they can tolerate exposure to low levels of oxygen. Okay? So, and we can test this using liquid thioglycate growth medium to identify the oxygen requirements of organisms. So the oxygen concentration is going to be higher up here and lower down here because we have a loose fitting cap. So a little bit of oxygen is getting in over here. Practically no oxygen is getting in down here. Okay, so obligate aerobes. Let's go through this. It's obligated to have oxygen. So where is the bacteria going to grow? Up here where the oxygen is. Okay, next one. Let's see if you got this. Obligate anaerobes. It's obligated not to have oxygen. Okay, so where is it going to grow? Down here. Facultative. It really likes to grow in an anaerobic condition, but it can grow either place. So you're going to see some more. It prefers anaerobic conditions, but it can grow this entire place, right? And aerotolerant aerobes are a little more spread out. So this is facultative anaerobes. This is aerotolerant anaerobes. There, if you want to see that. Okay. Nitrogen requirements. So it's the same thing with nitrogen. Anabolism often ceases due to insufficient nitrogen. Nitrogen acquired from organic and is acquired from organic and inorganic nutrients. All cells recycle nitrogen for amino acid and nucleotide production. And nitrogen fixation by certain bacteria is essential to life on Earth. So we need, for example, there are symbiotic nitrogen fixing bacteria on plants that actually fix the nitrogen, or they break down the nitrogen from the soil. So they take nitrogen from the soil and they break it down for the plant. That's a bacteria, a symbiotic relationship, mycorrhiza on the root nodules of plants that break down nitrogen for them so they can consume it. So that is pretty essential for a lot of plants. Trace elements and growth factors, sulfur, phosphorus. So there are others, and you will see this as we continue.